Welcome to The Deep Dive, the show where we take a stack of fascinating sources, cut through the noise, and really try to extract the most important nuggets of knowledge and insight for you. Today, we're diving deep into a truly intriguing subject, a small but mighty molecule known as SS-31. It's also referred to as Peptide. Now, this isn't exactly a household name, probably, but it's really turning heads in the serious research community. To set the scene, I want you to just imagine a microscopic key, right? Custom designed to unlock and enhance the performance of your cell's power plants, those tiny energy factories we call mitochondria.
Right, the powerhouses.
Exactly. SS-31 is exactly that kind of experimental molecule, holding the potential to help these vital cellular engines run more smoothly and, well, efficiently. That's the captivating world we're about to explore. To guide us through this complex yet utterly compelling science, I am absolutely delighted to be joined by Dr. Jason Miller. Dr. Miller is a clinical researcher specializing in regenerative medicine, bringing with him, well, extensive experience in understanding tissue repair and cellular health. He's here to help us unpack exactly what SS-31 is, how it actually functions at a fundamental level within our cells, why it's generating so much interest among researchers, and, what the current science tells us about its potential. We'll keep our discussion clear, conversational, and accessible for all of you listening. Dr. Miller, it's a real pleasure to have you with us.
Thank you, Alexa. It's great to be here and dive into such an exciting area of research.
Let's start right at the beginning, shall we? For those of us who might be hearing about this for the very first time, can you tell us, maybe in simple terms, what exactly is SS-31?
Absolutely. So SS-31, or Peptide, as it's often known in scientific literature, is fundamentally a small synthetic peptide. And when I say small, I truly mean it's incredibly compact. It's made up of just 4 amino acids.
Just 4? Wow.
Yeah, just 4. Now, for context, most proteins and even many peptides are much, much longer, consisting of dozens or even hundreds of amino acids. So 4 is, well, remarkably tiny. But don't let its size mislead you. It possesses a very unusual and highly specialized design that grants it some truly unique capabilities.
Okay. Like what?
Well, what makes SS-31 particularly fascinating and a significant focus of research boils down to a few key actions it performs. Firstly, it has this remarkable ability to easily pass through cell membranes.
Right.
This might sound trivial, maybe, but it's actually a huge hurdle for many therapeutic molecules.
Yeah.
Getting inside a cell is often the first and sometimes the biggest challenge. SS-31 navigates this with surprising ease. Then, once it's inside the cell, it doesn't just wander aimlessly. It's incredibly targeted. It specifically accumulates within the mitochondria, those cellular powerhouses you mentioned earlier.
So it goes right there.
Exactly. This selectivity is crucial because it means SS-31 goes directly to the site where it's believed to exert its beneficial effects. And once it's inside the mitochondria, it performs another very distinctive action. It strongly binds to a particular lipid called cardiolipin.
Cardiolipin.
Yes, cardiolipin. This binding action is quite specific and really central to its proposed mechanism.
Okay, so let me try to get this straight for myself and for everyone listening using an analogy.
Sure.
If we picture our mitochondria as these bustling little power plants within our cells, right, constantly generating all the energy our bodies need.
Precisely.
The cardiolipin, this lipid you mentioned, sounds like it's a special kind of structural component, almost like the essential bolts or maybe the specialized framework that holds all the power plant's intricate machinery tightly in place, keeping it stable and allowing it to operate efficiently.
Yeah.
And SS-31 then would be like a precision mechanic, sort of coming in to protect those critical bolts or that framework from rusting, maybe, from getting damaged or weakening, which in turn helps the entire power plant stay intact and, humming along. Did I capture the essence of that correctly?
That is an exceptionally vivid and, accurate analogy, Alexa. You've truly hit on the core concept there.
Very good.
Cardiolipin is indeed a very special component of the inner mitochondrial membrane. It's absolutely essential for maintaining the stability and, well, the proper function of the entire energy-producing machinery within the cell. It's not just any lipid. Its unique structure helps to organize the protein complexes that are responsible for electron transport, which is, the final crucial step in energy production.
Right.
So without properly functioning cardiolipin, the mitochondrial power plant becomes disorganized and, frankly, inefficient.
I see.
So yes, by protecting cardiolipin from something we call oxidative stress, which we can definitely delve into in more detail shortly, SS-31 appears to help mitochondria maintain their crucial structure and function.
Okay.
This is vital, especially when cells are under duress, maybe experiencing stress, or simply as they age. It's like reinforcing the most vital, delicate, and often overlooked components of that cellular power plant, ensuring its longevity and output.
That makes a lot of sense, this idea of a targeted architect or protector for these vital components. But this immediately leads me to wonder, why are researchers so incredibly focused on mitochondria in the first place? Beyond just energy, what makes them so central to our overall health and, well, so fascinating to the scientific community?
It's a foundational question, really, and the answer truly underscores the profound importance of mitochondria beyond just their, well-known role in producing ATP or cellular energy currency.
Right.
While energy production is undeniably critical, mitochondria are actually at the nexus of a remarkable array of other fundamental cellular processes.
Such as?
Well, for instance, they play a crucial role in programmed cell death, also known as apoptosis.
Apoptosis. Heard of that.
Yeah. And this isn't some, morbid process. It's vital for healthy tissue turnover, removing damaged or old cells to make way for new ones, and even as a defense mechanism against potentially cancerous cells. Mitochondria are like the cell's carefully calibrated self-destruct mechanism, ensuring quality control.
Wow, okay.
They're also deeply involved in various signaling pathways, acting as communication hubs that allow cells to respond to their environment and coordinate their activities. And perhaps more surprisingly, recent research has highlighted their significant role in immunity, helping the body fight off infections and regulate inflammation.
Immunity, too. I didn't know that.
Yes. So to call them just power plants is, frankly, an understatement. They are truly integrated command centers for many critical functions within the cell.
Integrated command centers. I like that.
Because of this incredible centrality, when mitochondria don't function optimally, the consequences aren't confined to just, feeling tired. They can be profound and widespread, particularly affecting tissues that have exceptionally high energy demands.
Right, that makes sense.
Think about it. Your muscles, which are constantly contracting and relaxing. Your brain, always processing information, firing neurons. Your heart, which never stops pumping blood. And your eyes, specifically the retina, continuously processing light and sending signals.
All incredibly busy tissues.
Exactly. These are all tissues that are heavily reliant on highly efficient, healthy mitochondrial function. When that efficiency falters, these tissues are among the first to suffer, leading to, a cascade of problems. This is precisely why scientists across so many disciplines are so invested in supporting mitochondrial health. They increasingly suspect, and accumulating research supports this, that improving or maintaining mitochondrial health could have significant implications for a broad array of conditions in areas of scientific interest. This includes understanding and potentially mitigating aspects of aging, addressing chronic fatigue syndromes, influencing various aspects of cardiovascular health, and even exploring new avenues for neurodegeneration, like in Parkinson's or Alzheimer's research. It's truly a foundational element of overall cellular and, systemic well-being.
So if these mitochondrial power plants start to struggle, it's not just one light bulb that dims, but the entire system starts to falter. It really is that fundamental to almost everything.
Exactly. Imagine a city where the central power grid starts to become unreliable. It affects everything, right? Transportation, communication, not just the lights in individual homes. Mitochondrial health is much the same for the body.
That's a really clear way to put it. Now that brings us back to something you mentioned earlier. Oxidative damage or oxidative stress. I often hear that term. And while it sounds bad, it can still feel a little abstract for many of us. Could you break down what oxidative damage truly is in simple terms and why it poses such a significant problem for our cells and specifically for those vulnerable mitochondria?
Absolutely. Oxidative stress is essentially an imbalance within our cells. To understand it, let's go back to our power plant analogy.
Okay.
As mitochondria work tirelessly to generate energy, it's a bit like a combustion process, right? And just like any combustion, there are natural byproducts. In this case, our cells naturally generate what are called reactive oxygen species, or ROS.
ROS, okay.
You can think of these as tiny, highly reactive sparks or embers that are an unavoidable part of the energy production process.
Little sparks.
Yeah.
Yeah.
Now, in a healthy, robust cell, these sparks are usually managed very effectively. The cell has its own intricate array of internal firefighting mechanisms, enzymes, and antioxidant molecules that quickly neutralize these reactive species before they can cause any harm.
Yeah.
It's a finely tuned system.
So the body has ways to handle them normally.
Exactly. But under certain conditions, this delicate balance can be disrupted. This happens when the production of these sparks dramatically increases, or when the cell's firefighting capabilities become overwhelmed or weakened, perhaps due to chronic stress, illness, or, well, simply the natural process of aging.
Right.
When this imbalance occurs, these reactive oxygen species begin to accumulate, and instead of being quickly put out, they start to damage nearby cellular structures, much like unchecked embers could scorch and damage anything too close to them.
Like cellular damage, literally.
Precisely. And this is exactly where cardiolipin, that unique lipid we discussed, comes back into the picture as a particularly vulnerable target.
Ah, because it's right there.
It's right there. And its specific chemical structure makes it highly susceptible to this oxidative damage. When cardiolipin gets hit by these sparks, its structure changes, its integrity is compromised, and as we discussed, this can destabilize the entire electron transport chain and the broader mitochondrial energy-producing machinery.
Okay.
This leads to a vicious cycle. Damaged mitochondria produce even more reactive oxygen species, further exacerbating the damage.
Oh, wow, a downward spiral.
Kind of, yes. So what research suggests SS-31 does is truly remarkable. It appears to help reduce the generation of those initial sparks, and critically, it helps shield cardiolipin from this damaging oxidative attack. The ultimate aim is to help preserve the mitochondria's ability to output energy effectively, maintaining its structural integrity and therefore its function.
So SS-31 isn't just dealing with the aftermath. It's actively protecting the vulnerable heart of the system. In our analogy, it's like a tiny, extremely precise fire blanket that covers and safeguards the most sensitive points of the mitochondrial power plant from those damaging sparks.
That's a very apt way to visualize it, yes. It's about targeted, proactive protection right at the source of potential damage.
That clarifies a lot. Now,
Sure.
I've also read that SS-31 has antioxidant properties, which naturally makes me wonder, is it just another antioxidant, like, the vitamins we might find in a supplement? Or is there something fundamentally different about its approach and why it's getting so much scientific attention?
That's a truly insightful question, and it gets to a key distinction in how different compounds function within our cells. While SS-31 certainly exhibits antioxidant-like effects, it is in fact quite different from what we typically categorize as general antioxidants.
How so?
Well, let's consider common dietary antioxidants like vitamin C or vitamin D. These are often referred to as general or scavenging antioxidants. They circulate throughout the entire cell or in specific compartments, ready to neutralize free radicals and ROS wherever they encounter them.
Right. They float around.
They're like a broad-spectrum cleaning crew mopping up spills wherever they occur. They're essential, absolutely, but their action is relatively nonspecific in terms of location within the cell.
Okay.
SS-31, however, operates with a much higher degree of specificity and precision, almost like a specialized unit. As we've highlighted, it doesn't just float around the entire cell. Its unique structure allows it to accumulate specifically inside the mitochondria.
Right, the targeted accumulation.
Exactly. This is a critical point because mitochondria are the primary sites where the vast majority of reactive oxygen species, those sparks, are generated in the highest concentrations within the cell. So SS-31 goes directly to the heart of the problem, the busiest production line of these potentially damaging byproducts.
Makes sense.
But its mechanism goes even further than just being in the right place. Instead of merely neutralizing free radicals after they've formed, which is what many antioxidants primarily do research, suggests SS-31 works by directly helping to maintain the integrity and proper structure of key lipids like cardiolipin and specific proteins within the inner mitochondrial membrane. By preserving these crucial structures, it helps ensure that the mitochondrial machinery, particularly the electron transport chain, runs more smoothly and efficiently in the first place. The prevailing idea is that if the system is running optimally, with all its components in their correct configuration, it may actually produce fewer radicals and damaging sparks to begin with.
I see.
So it's a more upstream, preventative, and structural approach rather than just a reactive cleanup.
So it's not just cleaning up the mess after it happens. It's actually helping to prevent the mess from happening, or at least significantly reducing its initial production by ensuring the machinery stays in top condition.
Yeah.
That's a fundamental difference and sounds much more, well, strategic.
Precisely. It represents a more upstream intervention, aiming to optimize the system's function and structure, thereby reducing the cause of oxidative stress, rather than just reacting to its symptoms or byproducts. It's a testament to, well, the elegant solutions biology comes up with, and what modern chemistry can sometimes mimic.
With such a targeted and seemingly strategic action, it's really no wonder researchers are so incredibly interested in SS-31. Let's dig into what they've actually found. What are some of the key areas of study where SS-31 is being investigated, and what kind of signals or results are they observing in these research settings?
There are indeed several fascinating and diverse areas where SS-31 is being explored. And while the specific applications might seem disparate at first glance, the common unifying thread, as we've consistently noted, is always the pivotal role of mitochondria and their health.
Right, it always comes back to the mitochondria.
Exactly. So one prominent and actively researched area is skeletal muscle studies. Imagine researchers conducting experiments on aged animal models, typically mice, which naturally experience a decline in muscle strength, endurance, and overall mitochondrial function as they age.
Okay, like in older mice.
Yes. In these preclinical studies, SS-31 has shown some truly intriguing results. For instance, researchers have observed signs of improved endurance, maybe measured by how long the animals could run on a treadmill or how quickly they fatigued, and a significant reduction in muscular fatigue compared to untreated aged controls.
Wow.
The hypothesis here is that by supporting mitochondrial efficiency within their muscle cells, SS-31 enables these muscles to sustain activity longer and perform better without succumbing to exhaustion as quickly. They might look at markers like ATP production rates, oxygen consumption, or even the accumulation of metabolic byproducts that signal fatigue.
So direct performance improvement in aged animals.
It seems so in those models. But it's important to note a key nuance that really highlights SS-31's potential therapeutic window. These beneficial effects were generally not seen in young, healthy mice.
Oh, interesting. Why is that significant?
Well, it suggests that SS-31 isn't necessarily boosting already optimal function to, super physiological levels. Instead, it appears to act specifically when mitochondrial problems are already present, helping to restore or preserve function in a compromised system.
Okay, so it's targeted at dysfunction.
That seems to be the case. This is crucial for understanding its potential as a targeted intervention for age-related or disease-related decline. Of course, and I have to stress this, these are preclinical findings from animal models. We cannot automatically assume they would apply directly to people, but they provide a strong basis for further investigation.
Right, right. Animal studies are a starting point. So it seems to have a restorative or protective effect rather than a universal performance-enhancing one, which is an important distinction. What about other high-energy demand tissues? I imagine the heart would be a prime candidate for this kind of research.
Precisely. That brings us to heart research, which is definitely an area of significant focus.
Okay.
The heart is, without question, a muscle with incredibly high continuous energy demands. It never rests.
Never.
SS-31 has been extensively tested in various animal models of cardiovascular disease, including heart failure and something called ischemia-reperfusion injury.
Ischemia, that's a lack of blood flow, right?
Exactly. Ischemia is a condition where blood flow to the heart is restricted, leading to oxygen deprivation. And reperfusion injury occurs when blood flow is restored, which ironically can cause further damage due to a burst of those reactive oxygen species we talked about.
Okay, so what did they find in those heart models?
Well, in these models, the results have been pretty compelling. Studies suggested that SS-31 might help preserve heart muscle function under stress, often quantified by metrics like ejection fraction. That's the percentage of blood pumped out with each beat or contractility.
Better at pumping?
Potentially, yes. Researchers linked SS-31 treatment with observations of significantly smaller areas of damage measured by infarct size after induced heart stress in these models. Furthermore, in models of aging hearts, which often exhibit reduced efficiency, SS-31 was associated with improved relaxation and contraction, vital aspects of a healthy cardiac cycle.
That sounds promising. What about in humans?
Moving into human studies, there have been early small clinical trials. In these trials, short-term SS-31 infusions were indeed associated with signals of improved heart pumping efficiency in some patients with heart failure.
Ah, so there are human trials.
Yes, but, and this is a critical caveat, these trials were small and very preliminary. While encouraging, we absolutely need much larger, long-term, and rigorously designed studies to confirm whether these observed effects are truly robust, statistically significant, and ultimately translate into clinically meaningful benefits for human patients in their daily lives.
Right. So promising hints and signals in heart health. But as you said, the journey from initial findings to conclusive treatment is definitely a long one. What about another critical high-energy tissue like the eyes? You mentioned those earlier.
That's an excellent segue, yes. The eyes, particularly the retina, are another major focus. The retina, the light-sensitive tissue at the back of your eye, is one of the most metabolically active and energy-demanding tissues in the entire human body.
I wouldn't have guessed that, but it makes sense.
It does. Its cells, especially the photoreceptors, require a constant high supply of efficient energy to process visual information and rapidly regenerate the molecules involved in sight. This makes the retina particularly vulnerable to mitochondrial dysfunction and oxidative stress, which are implicated in numerous blinding diseases.
Like macular degeneration?
Exactly. Researchers have tested SS-31 in animal models of various severe eye conditions, most notably age-related macular degeneration, AMD, which is a leading cause of vision loss in older adults, but also in models of diabetic retinopathy and glaucoma.
Okay. And the results there.
In these preclinical studies, scientists observed several positive signals. They noted improvements in mitochondrial structure within retinal cells, which might involve examining electron micrographs for, better organized cristae, the inner folds where energy is actually made.
Better structure inside the mitochondria.
Right. There was also a measurable reduction in oxidative stress, that's cellular rusting, within these vital retinal cells. Furthermore, studies showed evidence of the preservation of overall retinal structure, which is absolutely critical for maintaining vision, and in some models, improved visual responses, often assessed through something called electroretinography, in treated animals compared to untreated controls.
Improved vision responses in animals. Wow.
Yes, but again, these findings are certainly intriguing and provide strong rationale for human trials. As with the heart research, human clinical trials are still absolutely necessary to determine if such observed effects could be replicated and provide therapeutic benefit for people suffering from these debilitating eye diseases.
Understood. Preclinical promise needing human confirmation. That's truly remarkable, though. Muscles, the heart, and vision, all areas deeply affected by energy demands. Sounds like the broader context of aging itself is something that keeps coming up as a central theme in this research. Has SS-31 been studied more broadly in the context of general age-related decline, not just specific diseases?
Yes, very much so. The concept of aging is indeed a central and overarching theme in SS-31 research. Mitochondrial dysfunction is now widely recognized as a hallmark of aging, contributing to the general decline in physiological function that occurs as we get older.
A hallmark of aging, yeah.
Numerous animal studies have specifically investigated SS-31's effects in the context of biological aging. In aged mice, for example, SS-31 was associated with significantly better exercise tolerance and a range of markers indicating improved mitochondrial function.
We talked about the exercise tolerance, yeah.
Right. And these markers could include increased oxygen consumption rates, enhanced ATP production, reduced mitochondrial reactive oxygen species emission, and even improvements in mitochondrial biogenesis. That's the process by which new mitochondria are actually formed.
Making new mitochondria.
Potentially stimulating that process, yes. What's particularly striking in some of these studies is the observation that older mice receiving SS-31 performed more like younger, healthier ones in terms of their physical capacity, their metabolic profiles, and their cellular energy markers. They showed increased vitality, sustained activity longer, and recovered more effectively.
Older mice performing like younger ones.
In some aspects, yes. Now, it is absolutely vital to reiterate, as we have been, that these are animal studies.
Right.
We cannot assume the exact same results or effects would directly apply to humans. Animal models, while invaluable, are not perfect replicas of human biology. However, what these studies do provide is compelling evidence for the potential pathways and possibilities when mitochondrial health, which naturally declines with age, is specifically targeted and supported. They help us understand the mechanisms by which aging might be modulated at a cellular level.
Wow. So, in a way, it seemed to offer those older animals some of their cellular energy back, enabling them to function at a higher level, almost pushing back against some aspects of age-related decline.
That's certainly one way researchers have described it, yes. It speaks to the potential impact of addressing a foundational element of cellular aging, offering a glimpse into how maintaining mitochondrial vitality could influence our health span, not just lifespan.
Health span. That's a great term.
And beyond these specific high-demand tissues, there are also other broad research areas where SS31 is being actively investigated, again, always with that mitochondrial link.
Okay, like what else?
This includes preclinical investigations into conditions like acute kidney injury, particularly in diabetic mouse models, where mitochondrial stress plays a significant role in damage and recovery. Researchers are also exploring various neurodegenerative models, such as those for Parkinson's disease, where dysfunctional mitochondria are strongly implicated in neuronal cell death and disease progression.
Parkinson's, too.
Yes, and furthermore, there's interest in its potential role in general age-related cognitive decline, with some studies looking at improvements in memory-related tasks in aging animals.
Memories as well. It really covers a lot.
It does, but the unifying principle across all these diverse research avenues is clear: the fundamental role of mitochondria. The underlying hypothesis that connects all of these studies is that if you can improve or protect mitochondrial function, thereby enhancing cellular energy production and reducing oxidative damage, then a wide array of tissues and physiological systems may benefit. It highlights just how central mitochondrial health is to the proper functioning of virtually every cell and organ in the body.
It's truly impressive how many different physiological systems are being explored in relation to this one small peptide. That naturally makes me wonder about the timeline for this research. How far along is it, specifically regarding clinical trials in people? Are we seeing similar signals in humans as in these animal models?
That's the critical juncture for any promising compound, isn't it? Moving from animals to humans.
Ex-
And SS31 has indeed progressed into human clinical trials, which is a significant milestone that, frankly, many compounds never reach. It's been studied, particularly in patients with conditions like heart failure and certain eye conditions, based on those strong preclinical signals we discussed.
Okay, so what's the verdict from those human trials?
Well, the results from these human trials so far have been what we term mixed.
Mixed. What does that mean exactly?
It means that while some studies have shown encouraging signals of improvement, for example, in exercise capacity in heart failure patients or certain biomarkers related to mitochondrial health, others have unfortunately not met their primary endpoints.
Ah, the main goal of the trial wasn't met in some cases.
Correct. A primary endpoint is the specific, predefined measure of success that a trial is designed to achieve. When a trial doesn't meet its primary endpoint, it means it didn't show a statistically significant improvement in that particular outcome for that specific group and design.
Okay. Why would the results be mixed like that?
Well, this is a common and often complex part of drug development. The reasons for mixed results can be varied. Sometimes it's due to the heterogeneity of the patient population, different stages of disease, other health issues. Sometimes it's the specific dosage or duration of treatment chosen. Other times it might relate to the sensitivity of the biomarkers measured, or even just differences in trial design.
So lots of factors.
Lots of factors. For instance, a drug that shows clear benefits in a very specific animal model might face challenges in a diverse human population where the disease pathology is maybe more complex or multifactorial.
Right. Humans are more complicated than lab mice.
In many ways, yes. But what researchers are learning from these mixed results is invaluable. It helps them refine future trial designs, maybe identify more specific patient populations who might benefit most, optimize dosages, and pinpoint the most effective endpoints to measure. So while SS31 remains an experimental compound, the fact that it has advanced into human trials and that it continues to be studied makes it one of the more closely examined and advanced peptides in development that specifically targets mitochondrial function. It's really a testament to the rigor of science that we meticulously evaluate these compounds at every stage.
So if I'm understanding correctly, it's very promising based on the preclinical work and some early human signals, but the results aren't yet conclusive enough to say this is definitely a widespread treatment. It's more of a complex picture, and scientists are still kind of refining their understanding.
That's a perfect summary, Alexa.
Exactly. We have good, encouraging signals that certainly warrant further investigation, but we definitely need more definitive data from larger, more robust, and perhaps highly targeted trials before we can draw firm conclusions about its clinical efficacy and, its broader application. The journey from the lab to widespread clinical use is often long and filled with these kinds of learning opportunities. Given all this potential and the excitement around this peptide, let me ask a more practical question that I'm sure many listeners might be wondering about. Since SS31 sounds so powerful and is a focus of so much research, is it something people can just, buy and use right now? Is it available to the public?
No, absolutely not. That's a profoundly important point to clarify, and I really appreciate you asking that directly.
Sure.
SS-31 is, at this stage, still an entirely experimental compound. It is being studied rigorously within controlled clinical trials, and it is currently available only for research purposes, specifically within these highly regulated scientific settings.
Only for research. Got it.
It is not approved for general use by the public in any country, nor is it meant for human consumption or self-administration outside of these strictly regulated research environments. The safety profile for broad human application, optimal dosages, and long-term effectiveness are still very much under active investigation. It's absolutely crucial that people understand this distinction between a promising research compound and an approved therapeutic agent. Any claims of its availability for personal use should be viewed with extreme skepticism.
Okay, good to clarify that point unequivocally. So we're talking strictly about the realm of scientific research and development here, not something for personal use at this time.
That's correct. It's purely in the domain of scientific inquiry, careful development, and stringent clinical evaluation at this point.
With so many different approaches to health and wellness out there, and even other compounds that aim to target mitochondria, if you had to summarize, Dr. Miller, what truly makes SS-31 stand out in the current research landscape? What's its unique selling proposition, so to speak, that keeps researchers so engaged?
Its specificity. Without a doubt, I'd say its specificity is its truly distinguishing characteristic. When we look at the landscape of compounds being investigated for mitochondrial health, very few possess the unique combination of abilities that SS-31 demonstrates.
Specificity. How so again?
Well, firstly, there's its capacity to effectively cross cell membranes. As we said, that's a formidable barrier for many molecules, but SS-31 does it efficiently. Secondly, once inside the cell, its ability to then selectively accumulate within the mitochondria is remarkable.
Right, it goes right there.
It goes right there. This isn't a broad spectrum effect. It's a targeted delivery right to the cellular compartment where the energy production and consequently oxidative stress are highest. This targeted accumulation means less off-target activity elsewhere in the cell, making it a potentially cleaner and more precise intervention.
Right.
But SS-31 takes it a step further still. Once inside the mitochondria, it doesn't just broadly act. It directly and strongly binds to cardiolipin, that unique essential lipid we discussed earlier.
The binding to cardiolipin.
Yes. This direct, targeted interaction with cardiolipin, helping to stabilize it and protect it from oxidative damage, is also quite rare and represents a very valuable and specific mechanism of action from a research perspective. This isn't just a general antioxidant or some nonspecific cellular enhancer, it's more like a precision instrument. This combination of highly efficient mitochondrial entry and specific cardiolipin binding makes SS-31 quite unique and a fascinating focus for research into mitochondrial function and dysfunction.
So it really is like a precision tool, custom designed for these crucial mitochondrial components, rather than, say, a blunt instrument. That makes its continued study seem even more compelling.
Yes, that's an excellent way to put it. It seems designed to address a very specific and fundamental problem at a very specific location within the cell.
That leads me to my final question for you, Dr. Miller. Looking ahead, what excites you most about where SS-31 research could potentially go? What's the biggest potential you see on the horizon for this peptide, even if it's still down the road?
Well, what truly excites me, and I think many of my colleagues in the research community, is the overarching potential applications in the broader context of aging.
Aging again.
Yes. As we've discussed, mitochondrial decline is recognized as a fundamental hallmark of the aging process, contributing significantly to a vast array of age-related conditions, from sarcopenia, which is muscle loss, to cognitive decline, cardiovascular issues, sensory impairment. The list goes on.
It touches almost everything.
It really does. So if researchers can find ways to effectively slow, mitigate, or maybe even reverse aspects of that mitochondrial decline, that could have profound and broad implications. It wouldn't just be about extending life, but fundamentally about influencing many of the conditions we currently simply associate with getting older. Imagine the potential for maintaining vigor, cognitive clarity, and physical capacity much later into life.
Maintaining quality of life, essentially.
Precisely. There's also a strong and continued interest in its role in eye health, which I personally find particularly compelling. Given that the retina is one of the most energy-demanding tissues in the body and incredibly vulnerable to mitochondrial dysfunction and oxidative stress,
Yeah.
the idea that a targeted intervention like SS-31 could protect these vital cells and potentially preserve vision as we age, or in the face of diseases like macular degeneration, is a very powerful and hopeful avenue of research. Losing one sight dramatically impacts quality of life, and any progress here would be truly monumental.
Absolutely.
So really, it's not just about longevity in the abstract, but deeply about enhancing the quality of life as we age, supporting our muscles to keep us mobile, our hearts to pump efficiently, and maybe even our vision to remain sharp. The goal is to allow individuals to maintain independence, engagement, and a high functional capacity throughout their later years. That's the broader health span impact researchers are hoping to understand and, ultimately perhaps achieve one day.
That's a truly thought-provoking vision, Dr. Miller, enhancing our health span right alongside our lifespan. Dr. Jason Miller, this has been an incredibly insightful and fascinating deep dive into SS-31. Thank you so much for sharing your profound knowledge and making such complex science so accessible and really engaging for all of us. It's been a genuine pleasure.
The pleasure was all mine, Alexa. It's always a privilege to discuss these exciting developments and where the science might be headed.
And to you, our amazing listeners, thank you for tuning in to this deep dive into the world of SS-31. We hope you feel a little more well-informed, perhaps, like me, genuinely curious about the incredible science happening all around us. If you enjoyed today's deep dive, don't forget to subscribe to The Deep Dive wherever you get your podcasts so you don't miss any of our future explorations. Before we go, just a quick but essential disclaimer. The mentioned products and peptides are not intended to refer to any peptides or products sold by Power Peptides. Products sold are strictly for research purposes only and not intended for human consumption. Take care, everyone, and we'll see you in the next deep dive.